Morning, everyone. I will be updating you all on vegetation response in the Colorado River Delta following some environmental flow releases in 2014 and some smaller flow releases that are ongoing. I work for the Sonoran Institute. We're a nonprofit that's based in Tucson, Arizona. I'm also a graduate student at the University of Colorado at Boulder. And I'm glad Pat went ahead of me because I think some of his background information on vegetation dynamics are going to feed into my talk pretty well. So, there we go. Okay, the Colorado River Delta is located in northwestern Mexico at the mouth of the Colorado River. As you can see at the satellite image on the right here, it's largely an agricultural landscape today. Less than 100 years ago, it was an expanse of 2 million acres of riparian, estuarine, and marsh habitat. But today, it's less than 10% of that original size, and there's only a few remnant areas of riparian and marsh habitat remaining, which are on this map here. So if we look at flows to the Colorado River Delta over time, starting back in the 1800s, you can see flows were extremely high, and when the Glen Canyon Dam was completed in 1960, flows essentially stopped reaching the delta. And with the exception of flood flows in the 80s and 90s, the delta has not regularly received flows from the Colorado River since that time, which has led to extreme environmental degradation. However, despite this, the flood flows in the 80s and 90s did regenerate a lot of riparian habitat along the corridor, which kind of awakened the scientific community and the environmental community to the fact that this was a fairly resilient ecosystem, at that time at least, and that by providing environmental flows to this region, there was great potential for restoration. Which brings me to the passing of Minute 319. Minute 319 is essentially an amendment to the 1944 water treaty between the U.S. and Mexico. And in addition to other water management components, it dedicated environmental flows to the Colorado River Delta for the first time in history. And this is the first time environmental flows have been dedicated to a binational river in the world. So this is a landmark agreement. I just want to point that out. Good job, everyone. And the flows were dedicated to the delta in two different ways. So there was a pulse flow, which was intended to mimic the flood flows that used to go into the delta in the springtime. And that pulse flow already occurred in 2014 in the spring. And the total amount was 105,000 acre-feet. The other remaining third of the total flows going to the delta are in the form of base flows. These are low volume flows that are intended to maintain restored areas and target existing riparian vegetation. I should point out too that these flows are over the course of a 5-year term. That's the term of the minute, and once the 5-year term is up, there's going to be more negotiations to determine if flows will continue to be dedicated to the delta. So to put these environmental flows into perspective a little bit, the blue line is the average discharge from 1923 to 1935 to the delta. You can see our little pulse flow blip in 2014 here. So this graph is pointing out the fact that although these environmental flows were critical to the system, they were necessary, the magnitude of these flows looks a lot different than what the system has seen naturally. In addition to different magnitude of flood flows, there's other changes in conditions along the channel that since the 80s and 90s have dramatically altered as well. So for example, there's declining groundwater levels, particularly in the upper portion of the river near the border with the U.S. There's also channel obstructions due to the fact that the river hasn't flooded in a long time. And in addition, tamarisk is extremely dense along much of the river channel today. So when you add in these other factors, the question is, can these smaller magnitude environmental flows have some sort of effect that you would hope for from a larger natural flood flow that we saw, for example, in the 80s and 90s? This brings me to some of the research I'm going to present today, which was targeted at looking at the vegetation response to environmental flows, in addition to a number of restoration actions that were implemented as well. And in particular, are increasing levels of management required to promote native vegetation establishment? So this is kind of getting at Peter's question as well. What other actions are needed to get cottonwood and willow and other native species establishment? I'm going to present some results from two studies, the first of which is a small-scale study that was conducted in 2014 for my thesis research, and it was targeted in the Laguna Grande restoration area, which is about a 5-kilometer stretch of river in the central delta. And it was a controlled, replicated experiment, so I was able to do different things with my experiment on the smaller scale that the larger scale monitoring was not able to do. And then the second component I'm going to present is this larger corridor-wide monitoring that we're conducting as part of the Minute 319 monitoring efforts. And Pat Schafroth is a part of this as well. And really we're looking at the longer-term response of vegetation to the pulse flow and the base flow releases. So I couldn't resist. My slide was a little bare. Tale of two studies. So the first study, which is my thesis research, I used 4 different restoration treatments, and I applied them to different areas along this area, the restoration site Laguna Grande. The first treatment was a control, so there was nothing happened, no inundation. The second treatment was in areas that were inundated by the pulse flow and base flows. The third treatment were in areas that had been inundated, but we had cleared undesirable species before the release of the environmental flows. So we cleared tamarisk and arrowweed primarily. In the fourth treatment, we did all of this, and we also applied cottonwood and willow seed to these areas after the recession of flows. So I have increasing levels of management with each treatment. And my hypotheses were that due to the fact that the Colorado River in Mexico is in this altered state, increasing levels of management are required to get native seedling establishment. And so it follows that my T4, treatment 4, with the highest level of management will have greatest levels of native species establishment success, followed by T3, T2, and T1. Again, my study area was in Laguna Grande. It's located in the central delta region in Reach 4, and it's approximately a 5-kilometer stretch of river. There are existing stands of riparian habitat, but it's dominated by tamarisk. I chose this area because there's already ongoing restoration efforts in the region, and extensive clearing of arrowweed and tamarisk was being done before the pulse flow, so it was a good place to do the work. This is what my experimental design looked like. It was modeled after Cooper and Anderson did a study in 2012 on the Green River with controlled clearing in different areas along the river. So I had my 4 different treatments, and I included some photos here on the bottom. I don't know if you can see them, just to give you an idea of what each treatment kind of looked like. So my control areas were in kind of upland areas by the river. So you can see it's primarily tamarisk, some arrowweed. Treatment 2, which were areas that were inundated by the pulse flow. These were located right along the river and were densely vegetated, usually with a mixture of native and non-native species. My treatment 3 was typically along meander areas, so these were historic meanders along the Colorado River that had been cleared and the channels had been excavated and graded prior to the pulse flow release. So kind of a clean palette for little native seedlings to establish upon. And you can see that right here. And then treatment 4 was where we also applied the cottonwood and willow seed using a hydroseeder. So this is what it looks like from the air. You can see the extent to which clearing was done prior to the pulse flow. The main channel of the river goes through the middle here, and then these are some of those historic meanders. And this is during the pulse flow, so you can see that water levels have risen. And this is just kind of an idea of where my plots are located within the aerial photo. Okay, I'm not going to get too much into details on methods, but I did look at seedling count and cover for various species, primarily Fremont cottonwood, Gooding's willow, tamarisk species, Baccharis, Flukia odorata, and arrowweed, and then other species as well. And then in addition to looking at seedling count and cover class, I also monitored various environmental variables. As Pat was talking about, these are variables that affect seedling recruitment. There's a whole number of them, including ground cover, canopy closure, sediment deposition, soil moisture, soil salinity and texture, groundwater levels, and seed dispersal and abundance. So I have a bunch of data on all of these things. I'm only going to present on soil salinity since I have some pretty interesting results, and a little bit on groundwater as well. So here's some photos of initial establishment. These two photos, this one on the left and this one here, are from May. You can see some non-natives mixed in with the natives. So Flukia odorata and tamarisk, and then some cottonwood and willow, tamarisk, and this one is taken in June. You can see the seedlings are a little bit bigger. And now getting into results from my study. This is May 2014, so this is immediately after the pulse flow and really looking at initial establishment. So we have one thing you should notice that there's pretty high numbers going on here, a high count. Nothing established in the control, that's to be expected. In my T1, so areas that were inundated, I have a mixture of tamarisk, Flukia odorata, and Baccharis. So I should point out too, the species going down: cottonwood, willow, Baccharis, Flukia odorata, arrowweed, and tamarisk. So even in the inundated areas, the Flukia odorata and the Baccharis species, which are both natives, were able to establish even in this densely vegetated area, as well as tamarisk and a little bit of arrowweed. In the cleared inundated areas, we see a big spike in the count, so a lot more plants are establishing in these areas. And in addition to all of those other species, we have cottonwood and willow as well in smaller densities. And then in the cleared, inundated, and seeded treatment, we have a similar composition with slightly more cottonwood present as well. So if we look at seedling count after one growing season, so this is October 2014, seedlings have grown, many have died. Here's what we see. And I'll point out too that this count is much lower than the y-axis on the previous graph. So we did have quite a bit of seedling mortality, which happens with these pioneer species. Again, control, there's nothing present. In my inundated plots, I had a large number of Flukia odorata, some tamarisk, and arrowweed. In the cleared and inundated treatment, you see some little bit of cottonwood holding on and some willow, Baccharis, a lot of tamarisk, and similarly for the cleared, inundated, seeded, more cottonwood species in this treatment than in just the cleared and inundated treatment. If I pull out just the cottonwood and willow from those graphs, you can see some pretty interesting things. So my hypothesis held pretty much that the treatment 4 actually had the highest number of cottonwood and willow combined, followed by my treatment 3, treatment 2, and then the control had none. Well, treatment 2 and treatment 1 did not have any establishment. And so this graph shows dark blue is May cottonwood. So you can see, especially in the seeded areas, cottonwood had much higher establishment. I won't get into the details of why that's the case right now, but you can ask me questions later. And not the case for willow. So the dark green is willow in May, and you can see it has lower establishment in the seeded areas, but overall the seeded areas did have a higher number of cottonwood and willow combined. And if you are slightly alarmed by these large bars of purple, which is the tamarisk, I will point out here, show some photos of what's going on in the site. So here's our large bushy cottonwood. I think this was in October 2014. And you can see some smaller tamarisk below. So the idea is that given the ideal conditions, given shallow groundwater, given low soil salinity, cottonwood and willow can outcompete tamarisk, and work by Anna Scher and others has supported this. And so that's what these photos are demonstrating, is that these cottonwood and willow trees are kind of shooting up and taking off, and tamarisk there's a lot of them, but they're in the understory. Real quickly, I wanted to show some of my soil salinity results. I'm still working through this data, but these results are from my sites that were inundated. So all sites that got inundated by pulse flows and base flows are presented here. And what I saw was a mean decrease by 56% from March 2014 to October 2014. So that's a very significant decrease. So pointing out that the function of these environmental flows is not just to establish native seedlings, but it's also to reduce soil salinity, groundwater salinity as well. It's a very important factor for these river systems in the Southwest. And then briefly, our drawdown rates from the pulse flow were not ideal. They were decent. They could have been a little bit lower, and you could have gotten a higher seedling establishment. I also wanted to point out that we got some secondary inundation in some of the sites from the base flows. So pulse flow came, inundated the sites, seedlings established, and then base flows came and inundated the sites again, and they all died. So that's what we see in this photo, where we have Plukio dorata in the brown here, which is all dead, and we had some willow at the site. So that was one component that was kind of unintended. And then I want to bring it back out. So that's kind of the end of my study. And I want to bring it back out to the bigger picture, look at corridor-wide vegetation response that we're conducting monitoring with the Minute 319 vegetation team. And so I'm talking about vegetation monitoring that's occurring along reaches 1 through 5. And for this monitoring, we assessed vegetation along seedling transects. We had 21 in total. Again, looked at a suite of environmental variables to see how they affected seedling growth and survival. We also established transects in the Laguna Grande restoration area with the idea that we will compare establishment in these cleared areas versus establishment along the river as well. And I'm going to talk pretty briefly because I don't have much time, but looking at our conditions assessment for riparian species recruitment. These are all of the conditions that we set out that should be met in order for cottonwood and willow to become established. We used the monitoring results from our environmental variables to fill out this table. And from this, we found that Reach 1 and Reach 4 along the river had fairly favorable conditions for native species establishment, although there was very high competition in both of these sites due to the dense existing vegetation along the river. If we look at Reach 4 prepared sites, which means that they were cleared prior to inundation, so this is in the restoration area, we find that they meet almost all conditions. So Reach 4 prepared sites had the best conditions prior to the pulse flow release. And if we look at our results now and compare these to our conditions assessment, we see that that follows pretty well. So we get on the x-axis here, we have reaches 1 through 5, and then we have the Laguna Grande sites 1 through 3. We've just grouped our results into those areas. And you can see these are seedling frequencies, so the frequency of seedling presence along the transects. We have Gooding's willow, cottonwood, baccharis species, and tamarisk species. So you're looking for the blue, green, and the red, and you can see a lot in R1, so reach 1, and then a little bit of baccharis in reach 4, and not surprisingly, the highest frequencies are found in the restoration areas. So now extending out another year, so this is after 2 growing seasons. Again, we see some seedling mortality, but the trend is sort of holding strong. We still have quite a high frequency of Gooding's willow and Fremont cottonwood in the restoration areas, as well as some cottonwood in reach 1. So to conclude, and these conclusions are really based on my smaller scale study, as well as the corridor-wide study, and I think these conclusions hold true for both scales. Inundation from the pulse flows and base flows did promote native species establishment. However, clearing definitely helps a lot. And in the absence of these natural large magnitude flood flows that used to occur in the past, clearing is probably something that needs to happen in order to get cottonwood and willow establishment especially. In addition, a key function of these environmental flows is to reduce soil salinity, and in some cases in Laguna Grande restoration area, soil salinity was reduced to levels that were more favorable for native species establishment. And lastly, management of water levels is critical after the initial release. So if we had managed water levels better in the Laguna Grande area, perhaps we wouldn't have gotten secondary inundation, and we could better control the rates of decline to get optimal native seedling establishment. So with that, thank you very much, and I hope I have time for questions.
Thank you for a great presentation. One of the things that I think that you're seeing is there's an effect on seed longevity that actually happens in the Salicaceae with the willow and the cottonwood. So part of the reason you're seeing a seasonal effect is probably salts and scouring, the removal of salt through flooding. But you also might be seeing an effect of the longevity or the lack thereof of the seed themselves. There's also a difference in the ability of a seed to withstand saline effects versus the ability of an actual established plant. And it's going to change with maturation over time. And I think you might be seeing some of that as well. But all in all, it really looks like it's on the right track.
Can you talk just a little bit about the salt dynamics, how it accumulates and for what reasons, and then how the flushing affects that?
So there's a few different things that occur. In the Delta particularly, there are areas in Reach 4 where the groundwater levels are fairly shallow, and this is caused by agricultural return flows. And so in many sites in that area, the groundwater is pretty close to the surface, and you're getting salts accumulating on surface soils due to the groundwater coming up and evaporating. In addition, there's areas where water is in the channel, but it's not going anywhere. It's sitting there. And so especially along the channel banks, you're getting extremely high salinities accumulating because the water is just evaporating and leaving all of the salts on the soil. So environmental flows they're critical for flushing out the whole system corridor wide.
Was there ever much clonal coyote willow in that system?
There's a bit, there's not a lot of water in the system anymore. There's only water present in Reach 1 and Reach 4, and there are some patches in Reach 1, pretty sparse, and then there's patches in our restoration site as well. But just due to the fact that there's not, the river is dry in many areas in Mexico, so there's just not a lot of water to support it currently.
Just to chime in on that, Karen, there's the a couple patches that were really rejuvenated. Can you talk about that just quickly, and Corey?
Right. Yeah. So there were some existing patches prior to the flow releases, and since the implementation of the pulse flows and the base flows, they've expanded dramatically. So we have huge patches now in our restoration sites that have just expanded naturally. So we're seeing a response.